Okay. So now we will see about SDS page. Are you able to see the um this one sharing? What you can see now? Sodium dodecyl sulfate. Okay. Okay. So today we are going to see about sodium dodecyl sulfate polyacrylamide gel electrophoresis. That is SDS page. Okay. So here we are going to see what are the different features of the SDS page, how it works, the principal preparation of the gel, and how the process of SDS page is taking place. And after the separation is done, how to visualize the protein and what are the applications, advantages, and disadvantages of SDS page. Okay. So what is SDS page? என்ன நினைக்கிறீங்க? What is it used for? SDS page is used for the separation of proteins, mainly proteins, but it can also be used for the separation of nucleic acids. Okay, but commercially, and would have heard a lot of things that SDS page is used mainly for the separation of proteins, whereas agrocell electrophoresis is used for the separation of nucleic acids. Okay, so this is what it is. It is widely used in biochemistry. So you would have heard by the latest techniques, no, molecular biology techniques. So it is used in biochemistry, separation and isolation techniques. It is used in forensics. It is used in genetics and also in microbiology, molecular biology. Okay. So Lamely system. So Lamely system is the system of sample. Okay, sample preparation. Lamely system of SDS page was first introduced in the 1970s. See, 70s they have introduced. So what is the principle? How do the protein get separated? Naturally, all the proteins have some charge. Proteins are made up of what? Proteins are made up of amino acids. Okay, so different proteins are made up of different amino acids in a particular sequence. Okay, so naturally they have some kind of charge because of the charge present in the amino acids and the proteins. So this charge can be attributed by the acidic amino acids or the basic amino acids, or when they are ionized, the proteins or the amino acids generally, whenever in which medium, or yesterday, as I told you yesterday, in which pH they are present, accordingly they can be ionized, so they can be made to charge. But here in SDS page electrophoresis, all the molecules are made to given a negative charge. Okay, are converted into negative charge, so that they can be separated in an electric field. So this is a polyacrylamide gel electrophoresis. So electrophoresis is a technique where charged molecules can be separated by allowing them to migrate in an electric field. Okay, this is how the principle is used in SDS page. Okay, not only SDS page, you have polyacrylamide gel electrophoresis also. The difference between polyacrylamide gel electrophoresis, that is page, and SDS page is that in the SDS page, okay, you give a total charge, a complete negative charge uh, to the molecule. Whereas in page, you don't give attribute any charge externally to the protein molecules. The protein molecules are allowed to separate depending upon its own electric charge. Okay, that is the main difference between the page and SDS page. So what is SDS? As you know, SDS we have seen the full form, sodium dodecyl sulfate. It is an anionic detergent. Okay, anionic means it is a negatively charged detergent. So when it is attached to the protein molecules, so how it gets attached to the protein molecules? One molecule of SDS can bind to two molecules of amino acids. Okay, one molecule of SDS can bind to two molecules of amino acids. So suppose a protein is made up of 10 amino acids. Okay, so 5 molecules of SDS can bind to them. Suppose a protein is made up of 50 amino acids. So totally 25 SDS molecules can bind to them. Okay. If the protein is 100 molecule, it is made up of 100 molecules of amino acids, then 50 number of SDS page can get attached to the protein. So which is a bulk protein? The one which is having 10 amino acids, or 100 amino acids. The one which is having 100 amino acids are bulkier in nature. Okay, so when they are attached with SDS, okay, they will carry a total net negative charge. Okay, so the net charge carried by protein made up of 10 amino acid is very small. Okay, it will carry only 5 charge, whereas that having 50 amino acids carry 25 charge. Okay, so those proteins having 100 amino acids, they carry 50 negative charge. So when they are allowed to migrate in an electric field, which will move faster? Which will move faster, tell me? The one having less number of charge or the one having larger number of charges? Are you there, students? You are listening? Yeah, tell me the answer. So out of three, I told you three examples. Out of these, which one will migrate faster? Louder, louder. கொஞ்சம் சத்தம் எனக்கு கேட்கல. லெஸ் சார்ஜ் இருக்கிறது வேகமா போகுமா இல்ல அதிகமா சார்ஜ் இருக்கிறது வேகமா போகுமா? So what do you mean by separation of proteins in an electric field? அதாவது எலக்ட்ரிக் ஃபீல்டில் ரெண்டு பக்கம் ஒரு ஆனோடு ஒரு கேத்தோடு வச்சிருப்பாங்க. வாட் இஸ் ஆனோட்? ஆனோட் இஸ் அ பாசிட்டிவ்லி சார்ஜ்ட் எலக்ட்ரோட் and cathode is a negatively charged electrode. So when charged molecules are allowed to migrate or travel in this electric field, so how they are allowed to travel? The protein molecule or the charged molecules will be allowed to travel in a gel, in a medium. Okay, so they will be attracted towards the oppositely charged electrode. Suppose positively charged cation will migrate towards the cathode, negatively charged anion will migrate towards the anode. Okay, this is how it is. So my question is, when molecules of different charges, say 5 charge, 5 all are negative charge, there is no positive. Okay, all the three are negatively charged, so they will all migrate towards the positively charged anode. Okay, but there is a difference in the rate of migration. So what is the difference in the rate of migration? I am asking you whether the minus 5 charge, minus 25 charge or minus 50 charge, which will migrate faster towards the anode? Which one will migrate faster towards the anode? The migration rate is faster for which molecule? Somebody told me the one which is having lesser number of charge. Is it correct?
Yes, ma'am.
No. The one which is having lesser number of charge will migrate slow. The one which is having maximum number of charge, say here it is minus 25, will migrate faster. Okay, எதுக்கு அதிகமான strength இருக்குங்க, அதிகமான number of charge இருக்கிற ப்ரோட்டீனுக்கு தான் it will migrate faster. Okay, so those protein molecules which have higher number of charges, here it is minus 25. Okay, that is minus 50. The protein molecule having 100 amino acid, when it is attached with SDS, 50 molecules of SDS will bind to this particular protein. I told you, one molecule of SDS will bind in between two amino acids. So when the protein is having 100 amino acids, 50 molecules of SDS will bind to it. Okay, so the total charge attributed to the protein by binding with SDS will be 50. Okay, similarly, depending upon the length or the length of amino acid or the molecular size of the amino acid, okay, the number of SDS will be based upon the number of amino acids. Okay, if the number of amino acid is 10, 5 SDS will be added. Okay, so the total charge will be minus 5. So when they are allowed to migrate in an electric field, those which are having higher number of charges will migrate faster through the medium towards the anode, whereas those which are having lesser number of charges will migrate slower in the medium. So you have to understand this. Purida, are you able to understand? Please tell me yes or no. Okay, so those molecules which are having higher number of charges migrate faster than the molecules which are having lower number of charges. The other molecules in between, they will have a, that is the migration rate will be in between the fastest and the slowest molecule. Okay, so with separation of proteins in an electric field based upon the charge they possess. This is the principle behind SDS page. So how do they migrate? Summa thanila pota migrate aguma. Thanila pota, it will not migrate. Okay, it should have a suitable medium through which the proteins to be separated should travel. Okay, so the medium should be either a semi-solid. Okay, it should be a semi-solid medium or a gel. Okay, so the protein molecules should be allowed to pass through the gel. So what is the nature of the gel? The gel is soft in nature, and it will allow, allow the protein molecules to migrate. It will have some pore size. Okay, so the pore size will be decided by the polymerization of the molecules. So here SDS page polyacrylamide gel electrophoresis, it is prepared by polymerizing the acrylamide and the bisacrylamide together. Okay, it is used by mixing the acrylamide and polyacrylamide. So here the acrylamide concentration will be more, and they will be forming horizontal polymer linkages, whereas the bisacrylamide is a dimer. Okay, that is, it is made by attaching two molecules of acrylamide. So they will be acting as a bridge between the two horizontal polymers. We will see the structure later. Okay, so they will be migrating through the gel. So this will allow the pore size. This will give a pore through which the proteins can migrate. So here, unlike gel electrophoresis, here the molecules migrate through the pores, but the separation will depend upon the charge. Okay, so totally the electrophoresis and the chromatography can be differentiated by how they move. Okay, based upon the charge or based upon the size. Some kind of molecules or some principles uses the difference in the sizes. For example, gel filtration chromatography. Here in this case, SDS page electrophoresis, the separation of molecule purely depends upon the difference in the charges, not based on the sizes. Okay, based upon the sizes, they will be able to move through the gel and they get trapped in the gel. Okay, so when they are allowed to pass through the gel in an acrylamide, there is a page vertical gel electrophoresis. Okay, you can see the smaller molecule, they may travel faster. Okay, through the pores. So according to that, the pore size should be determined. Okay, so the pore size and the concentration of the gel is very, very important. Okay, so the smaller molecules will move faster and they will attain the end of the gel, whereas the larger molecule will be present at the top of the gel. So this is very, very important. Now you can see this is the apparatus for gel electrophoresis. Okay, it contains a large trough or a tank where the buffer will be placed. Okay, well, the buffer will be kept, and you can see the vertical gel electrophoresis. This is the setup. Okay, there will be two gel plates. In between the two gel plates, the gel will be formed. Okay, and there will be two gels. One is the actual separation gel, and the other one on top is the stacking gel. Okay, on the stacking gel, some you can see wells are prepared. Okay, wells are prepared like this by using the comb, and in the wells the protein sample will be applied. Okay, the protein sample will be applied, either injected or pipetted into the wells. Okay, and here you can see this is the electrode. Okay, this is the negative electrode and it is on top, and the positive electrode anode will be down. So you can see the protein sample are allowed to react with some gel loading buffer. Okay, so they are allowed to react with the gel loading buffer. So they are made to they are some this gel loading buffer, okay, or sample loading buffer, it will contain certain compounds. So what are the compounds? It contains beta mercaptoethanol. Okay, and it contains SDS, it contains bromophenol blue, and also it contains glycerol. So what does it contain? It contains beta mercaptoethanol, it contains SDS, it contains bromophenol blue, and it contains glycerol. So what is the use of all these three? This sample buffer, which contains all this, will digest the sample. So protein is a macromolecule. Okay, it is a larger compound. So it has to be separated through the gel. So it has to undergo some kind of activation or some kind of change. Okay, so this beta mercaptoethanol, it will bind with the protein and it will denature the protein. Okay, denature the protein, it will unwind the protein. It will not lose its biological activity, but it will unwind the protein. So protein is a highly complex structure which is bound in the form of a round globular shape. Okay, so when beta mercaptoethanol, they will go and bind to the cysteine molecules. Okay, so the disulfide bridges will be broken. So what is the disulfide bridges? Disulfide bridges are used to connect the proteins, whether intrapeptide or the interpeptide linkages. Okay, by forming disulfide bridges between the two cysteine molecules. Okay, so this disulfide bridges is used for binding of the protein, binding of the protein, or you can say the folding of the protein. Okay, so when the bromo beta mercaptoethanol attacks the disulfide bridges, the disulfide bridges will be broken and the folding of the protein will get affected. So it becomes unfolded. Okay, it unfolds. Okay, that is what is happening. It unfolds and all the amino acids will be exposed. Right? Exposed now. Now the SDS will go and bind to the that is the unbound or the unwound proteins. Okay, so the SDS is an anionic detergent having negative charge. When this SDS goes and binds to the protein, as I told you, one SDS molecule will bind to two molecules of amino acid. So it will give a negative charge. Negative charge will be given to the protein, as I told you. So if a protein, depending upon the length of the protein, the number of SDS molecules will bind to half the length of the protein. That means if the protein has 20 amino acids, 10 molecules of SDS will be binding to the protein, giving a total charge of 10. Okay, similarly in a mixture of proteins there will be different size proteins. So depending upon the size and length of the protein and the number of amino acids present in the protein, the charge will be attributed to the protein. The larger the protein, the more the number of negative charge, and the smaller the protein, the lesser the number of negative charge. And the intermediate proteins will have, depending upon the number of amino acids, the SDS will be bind to them. Okay, now I have told you the function of beta mercaptoethanol SDS. The next one is bromophenol blue. The proteins are colorless. Okay, so when they are passing through the gel, it cannot be visible through the naked eye. So we have to find out how the protein moves through the gel, and the movement of the protein through the gel should be stopped, just like PLC and paper chromatography. It should be stopped before it reaches the end of the gel plate. Okay, otherwise what will happen, it will dissolve in the buffer. So before it enters the buffer, it should be stopped. So the movement of the protein should be visualized. So the bromophenol blue is a blue colored molecule which will bind to the protein, and it will allow us to visualize the protein as it moves through the gel. The next one, what I told you, is beta mercaptoethanol, SDS, bromophenol blue, and glycine. Okay, so, glycerol. So, what is the role of glycerol? Glycerol, when it is added to the protein molecule in the sample loading buffer, usually now it is called as the lamelli buffer. Lamelli buffer is a pre-prepared sample buffer. So, usually in our laboratory, mainly the students will add all these different ingredients to prepare the sample buffer. So the buffer will be taken to that SDS, bromophenol blue, and beta mercaptoethanol, glycine, everything will be weighed separately and it will be added to make the buffer. But nowadays, ready-made sample loading buffer or sample digesting buffer is available. That is called as the lamelli buffer. Okay, so the role of bromophenol blue is to identify the movement of the sample through the gel, whereas the role of glycerol is to give weight to the sample. Okay, it will add weight to the protein sample, so that when it moves from the well into the gel, it should have some weight, so that it sinks to the bottom of the gel and enters, all the protein should enter the gel at the same time. Okay, there should not be delay in entering the into the gel. Okay, there are different sizes of proteins. So all the different sizes of proteins may move at its own rate, okay, if it is not given some density. So when the glycerol binds to the protein, it will give some density to the protein, and it will sink to the bottom of the well first, and then it will allow the movement of the protein to the gel at the same rate. Initially, there is a stacking gel, so they will move into the stacking gel at the same rate, okay, till it reaches the end of the stacking gel. The concentration of the stacking gel is lesser than the concentration of the separating gel, and because of the difference in the concentration, the stacking gel has a bigger pore size when compared to the separating gel. So all the proteins will move through the stacking gel at the same rate till it reaches the separating gel. The separating gel, because of the increase in the concentration of the separating gel, okay, they have some difference in the migration depending upon the pore size and also the size of the protein. So the different proteins move at a different rate. Okay, so now what happens? They will all move towards the anode, but based upon the size, they get separated. Okay, based upon the size, they get separated. Though the bigger molecules possess larger negative charge, okay, the smaller molecules move faster through the gel pores of the gel, and they will occupy the end of the gel. Okay, so whereas the larger molecules, they will not be able to enter the gel as that of the smaller molecules. So the rate of migration will be slower because of the larger size. So they will be present as a band. So molecules of the same size will form a band, and they will move together and occupy the upper region of the gel. The smaller molecules will also move together as a family, and then they will occupy the lower portion of the gel. The intermediate molecules, the less smaller, அதை விட small, அதை விட small, அதை விட small, அதை விட small, they will occupy the other regions of the gel. So this is how they get separated. So as I told you, SDS coated large proteins. So proteins will be coated with SDS. Okay, so they will be given a negative charge because of the coating or binding of the SDS to the protein molecule, migrate slowly through the gel matrix, and small proteins migrate quickly through the matrix. Smaller ones, they can easily enter the pores, and they will occupy the lower edge of the gel. So near the well, the larger molecules, larger size molecule occupy here, and the intermediate molecules will occupy in the other regions. So as I told you, what is SDS? It is a negatively charged detergent called a sodium dodecyl sulfate. So it is used to denature and linearize the proteins. Okay, it is not used to denature. Beta mercaptoethanol is used to denature the protein. So beta mercaptoethanol will attack the disulfide bridges, and they will unwind the protein. Okay, the folded protein will be unfolded, and after it is unfolded by the beta mercaptoethanol, the SDS will come and bind and gives a negative charge to the protein. As I told you, one molecule per two molecules of amino acid. So it is differentiated into two systems. One is the continuous SDS page, and the other one is the discontinuous SDS page. Okay. So polyacrylamide is used to form a gel. Now, what is the gel used here? Stationary phase or the medium? You cannot say it is a stationary phase. The medium in which the proteins are separated. Okay. So it is called as the polyacrylamide gel. So the polyacrylamide gel, I'll tell you, it is made up of differing concentrations of acrylamide, increasing concentration of acrylamide, and a lesser concentration of bisacrylamide. Okay, I'll tell you how it is made. So the higher the concentration, the smaller. I told you, the size of the pores are decided by the concentration of the gel. When you increase the concentration, you will get the smaller pore size. When you decrease the concentration, you will get a larger pore size. So what is a continuous gel? The whole gel is made up of a single concentration. That is called as the continuous gel. Whereas a discontinuous gel is what is used in SDS page, where you have two different concentration of the gel. One is the separating gel, which is having a larger concentration. See, it is present at the bottom of the gel. The concentration will range from 5 to 15% of acrylamide. Whereas in the same gel plate itself, you stack a different gel, okay, by lessening the concentration of the gel. Here only 4% acrylamide is used. I told you that is because of the lesser concentration. the proteins will be able to migrate through the stacking gel and enter the separating gel at the same time. And this is aided by the presence of glycerol. The actual separation of the protein takes place in the separating gel, not in the stacking gel. So because you are using two different concentration of the gel, it is called as a discontinuous gel. So what is why polyacrylamide is used? It is chemically inert, it is electrically neutral and hydrophilic in nature, and it is transparent also. You can see the migration or separation of the proteins in the gel, and detection also can be possible by using a proper dye. So how the gel is prepared? Gel slab is prepared. The medium is prepared for running the electrophoresis. So you have a comb. Two plates are there. Okay, so the two plates are joined together, and it is separated by spacers on all the three sides. The spacers are used to separate and give a thickness to the gel. Okay, so they are, I'll tell you how the exact formation of the gel, how it is prepared. We will show you some pictures also, animation and a YouTube video. Okay, the plates are set up like this by using a spacer, and the gel is poured inside the in between the plates. Okay, and above you have to prepare two different types of gel. One is the separating gel and the other one is the stacking gel, and you have to place the comb, okay, which is used to make the well in the stacking gel. Okay, you can prepare the gel ahead 1 or 2 days back, and you can store it, or pre-prepared gels are also available in the market commercially. Okay, but it is a very good practice to prepare our own gels so that you can get an idea. Preparing a gel is not very easy. Okay, it will take a lot of time for initial users. Okay, it will I mean there will be a lot of trial and error basis to set up a gel. You have to use the actual concentration. And the gel is prepared by polymerization of acrylamide and bisacrylamide. Okay, for the polymerization, you have to use ammonium persulfate. So ammonium persulfate is an initiator for creating a free radical. Okay, so the free radical should be created first, initiated, and then the further polymerization can be catalyzed by the addition of TEMED. Okay, TEMED is used for the, so what will happen? This will result in the formation of a free radical. So more and more free radicals are formed and this will result in the polymerization of the acrylamide and bisacrylamide to form the gel. Okay, this is how the gel is formed. Okay, now after the gel is formed, now you have to add a different concentration of the gel is prepared for the separating and the stacking gel. That also we will deal with it later. So the after the preparation is over, it is allowed to set. Okay, it should not be if you take the spacer underneath, remove the spacer at the bottom, it should not leak. Okay, if it is leaking, then the gel is not set and you cannot run the sample inside the gel. So after the spacers are removed, the gel should be set. Okay. Now the sample, as I told you, it should be boiled for 5 minutes or to 10 minutes with using a proper sample loading buffer, which contains beta mercaptoethanol, SDS, glycerol, and bromophenol blue. So I told you what is the use of each and everything, and it will be poured. 20 microliter of the sample will be poured into the well. So whatever it is, whether you take 20 microliter, the sample concentration is very important. You have to take uniform concentration of the sample. Should be loaded into the well. Okay, if the concentration of the sample is not uniform, then the band formation will also be not uniform. Okay, so whether the volume of the sample is not important, the total volume of the sample should be 20 microliter that should be loaded into the well. Okay, so the volume of the sample can be mixed with the volume of some buffer and made equal to 20 microliter, but the concentration of the samples should be the same. Okay, it should be the same, say 550 microgram of the sample should be loaded exactly into each and every well. Okay, 50 microgram of the sample should be loaded into the well. Then the electrophoresis is run by attaching the electrode and switching on the current. Okay, so after that is done, the visualization of the band under UV light. Okay, so this you can use different methods by using Kumasi Brilliant Blue or silver staining. So Kumasi Brilliant Blue will detect around 100 nanogram of protein. Okay, whereas silver stain is more sensitive, where Even smaller amounts, much small, 0.1 to 1 nanogram protein also can be detected. Okay, so this is Kumasi brilliant is blue in color, and silver staining is something around black in color. Okay, this is the different staining. What you can see here, staining band with western blotting. Here, western blotting. Okay, western blotting. After the gel is taken, and it is the bands or the proteins are transferred onto a nitrocellulose membrane. So this is sustained, or you can say the detection of the protein is done by ELISA method with the western blotting, where western blotting is used to detect the proteins, whereas southern blotting is used to detect the nucleic acids. Okay, so this is the actual gel. This is not the gel. This is the nitrocellulose membrane. Okay, so this is a Kumasi brilliant blue. The remaining protein after transfer into the nitrocellulose membrane are stained again, and you can see the pattern of the protein that is undergone separation. You can match it with the protein ladder of different molecular weight, and you can see what is the molecular weight of the proteins. Okay, so you can see this is the ladder. Okay, ladder which shows the 20 kilodalton and 150 kilodalton, the largest protein. In between, you have different molecular weight proteins. So this is used as a reference marker for detecting the different molecular weight of the actual compound under separation. Okay, and this is the silver stain. So silver stain looks like this, brown color or black color stain, whereas Kumasi brilliant on a gel looks like this, and western blotting looks like a black color. Okay, it cannot be visualized through naked eye, but it can be visualized using a illuminator. Okay, so application. What is aluminum application used for purification of the sample protein? You to determine the molecular weight of the protein by using suitable ladders, and you can also identify the disulfide bonds present in the protein. You can also quantify the protein. Okay, what is the amount of protein also can be detected, and the blotting can be done. So blotting can be done, and you can find out what protein is present. By using a suitable antibody, you can blot the protein in a nitrocellulose membrane and find out what the nature of the protein. So you have advantages and disadvantages also. Proper use of the polyacrylamide gel is very, very important. So here, what are the advantages? Migration is proportional to the molecular weight. So depending upon the molecular weight of the protein, the migration will happen. The smaller molecular weight protein will migrate faster to the end of the thing, and the larger molecular weight protein will move slower because of the difference in the size. Okay, so here the because depending on the difference in the size, they get separated, and the principle is based upon the charge. They move only towards one direction. It does not move in both directions. All the molecules, protein molecules, are charged in negative, so they can move in the towards the anode. Okay, but the ones which are smaller migrate faster. The one which are larger migrate slower because of the size. Okay, so, but when the same thing is allowed to flow in a liquid medium, just like electrolysis. Okay, in electrolysis, when the gel is not used, only liquid is used, and the different proteins are separated. They will be separated based upon the charges. The higher the number of charges, the migration rate will be faster. Whereas the lesser the number of charges, migration rate will be slower when they are moving in a liquid medium. But because here, when they are moving in a gel medium, the gel will act as a filter. Okay, so depending upon the pore size, they will allow the smaller molecules to migrate faster through the gel, and allow the larger molecules to move slower because of the large size. So highly sensitive, separates a 2% difference in the mass. Even the difference of the protein is 2% difference also, they can be separated. The sample size required is very small amount, and the gel is very stable. The disadvantages are sometimes band resolution is very poor because of alkaline pH, and acrylamide, when you are using acrylamide, you should not touch it with bare hands. You should always use a gloves during the preparation of the acrylamide gel, or during the handling of the SDS page, or when you are disposing. You should not touch it with your hand. Acrylamide is a neurotoxin. Okay, so it is very, very dangerous, toxic. Now, as I told you, gel preparation is a trial and error basis. Okay, it is not easy for a first timer to prepare the gel. Okay, so it will by practice and experience you can prepare the gel. The polymerization is a little difficult. The appropriate use of TEMED, the APS, ammonium persulfate, is very important for the initiation of the gel setting and the polymerization. That is a little difficult to do, but by practice you can attain this. That is why nowadays, no, you cannot waste your time by preparing the gel, discarding the gel, etc. So what you should do is, you should always, people who are doing it for commercial purpose, where they have to show accurate result, and so they have to handle many number of samples, they will use a pre-prepared gel. Okay, and also it is costlier. So you can see, visualize. So this is the SDS page is a technique used to separate the proteins according to their molecular size through the gel. So this is what proteins are unfolded by the beta mercaptoethanol, and they migrate from cathode terminal end to the anode terminal end at different rates depending upon the size. So molecular weight also can be determined by using the this one, um, molecular ladders. So you are able to see this slide, this particular thing. What are you able to see now? தூங்கிட்டீங்களா? SDS polyacrylamide gel electrophoresis தெரியுதா? இப்போ என்ன தெரியுது? Objectives. Oh, உங்களுக்கு screen நல்லா தெரியுது இல்லையா? Now this is the procedure for separation of the protein using SDS page. So the objective of the study is the procedure what you can follow. Separation of protein fraction using SDS page. Okay, so SDS, what is it is sodium dodecyl sulfate polyacrylamide gel electrophoresis technique used in biochemistry, forensic, genetics, molecular biology to separate the and identify protein based upon the molecular weight. Okay, based upon the molecular weight or the size they can be separated, and also the molecular weight can be determined. Okay, principle, this is the principle you have to write. Okay, when you're asked to write the procedure, you have to write this. So what is SDS? It is a detergent which will be giving attributing negative charge to the protein. So what does it do? It will, the beta mercaptoethanol will bind to disrupt the disulfide bridge. Okay, and this SDS will bind to the hydrophobic regions. Okay, so it will, all non-covalent bonds will be disrupted, and the protein will get a net negative charge. Okay, that is the role of SDS. Now the role of by beta mercaptoethanol, instead of beta mercaptoethanol, DTT, dithiotretol can be used. So this, as I told you, it will be used to denature the disulfide linkages and opens the protein, unfolds the protein so that the SDS can bind. Okay, so the proteins will have a uniform net negative charge, and when they are subjected, passed through the gel and under the influence of an electric field, they will be migrating. So because all the molecules possess negative charge, they will all be migrating towards the anode in the medium. Okay, polyacrylate medium. So this is the beta mercaptoethanol. What does it do? The disulfide bridges are broken, so the protein, which is a globular protein, okay, it is highly folded, they undergo unfolding. Okay, now after the unfolding, you can see SDS, negative charge, this will bind. So beta mercaptoethanol used to disrupt the disulfide bridge, so the protein opens up and the SDS will bind to this. So the binding of the SDS depends upon the molecular weight or the size or the number of amino acids present in the protein. Okay, see here the size of the protein is small, so only two SDS is attached. Here the size of the protein is very large, so three number of SDS will be attached. So what happens? How the gel is prepared, and how the samples are coated. Okay, that is what is shown in this picture. Okay, so what is a polyacrylamide gel? As I told you, polyacrylamide gel is formed by the co-polymerization of acrylamide and bisacrylamide. So cross-linking அப்படின்னா என்னது? The one polymer will be in the horizontal direction, the other polymer will be in the vertical direction. So the just like a weaving of a cloth, they will be bound like this, no, horizontal and vertical direction. So the acrylamide forms the horizontal polymer, and bisacrylamide forms the vertical polymer. Okay, so for polymerization of this has to be converted into a free radical. The initiation of the formation of free radical is done by ammonium persulfate. Okay, so this will initiate, and the polymerization is continued by the addition of TEMED, which is acting as a catalyst for the polymerization. So this is the acrylamide. This is the structure of acrylamide. CH2 double bond CH2. When two molecules of acrylamide are joined together by disulfide bridge, it is called as the bisacrylamide. N dash methylene bisacrylamide. So as I told you, this is the polymerization, attaching of one monomer with each another to form a polymer. This is catalyzed by free radical formation. So the free radical formation is first initiated by APS, and then it is continued by TEMED. And this, you can see, this molecule is the bisacrylamide. So you can see number of acrylamide are more. See, 28.8 gram of acrylamide is used to prepare the gel, and only 0.8%, 0.8 grams of bisacrylamide is used. The percentage of bisacrylamide is very, very less when compared to the acrylamide. Okay, so this is how the bridges form. See, this is n times. You can see this is n times. So எங்கேயாவது ஒரு இடத்துல this cross linking bisacrylamide will be present. This will be, this polymer, horizontal polymer will be very, very large, whereas the bisacrylamide will be, the linking bridge will be very small number. You can see this is n number. Okay, so now this is the gel preparation. As I told you, this is about 80% of the gel will be made up of separation gel, discontinuous gel. Okay, so here the concentration is different and the pH is also different, whereas the stacking gel is made up of lesser concentration when compared to the separation gel, and the pH is also different. Here the pH is 6.8 and here the pH is 8.8, and the wells are prepared by using a comb. So the sample, how it is prepared, I told you, no. This is the lamellae buffer, or a sample loading buffer. It contains SDS buffer, tris HCl buffer at pH 6.8, glycerol, beta mercaptoethanol, and bromophenol blue. I told you the use of all this. So the 40 microgram of protein sample is boiled with the 10 microliter of this buffer. Okay, so it contains the medium buffer tris HCl. I told you what is the role of SDS. I told you what is the role of beta mercaptoethanol, bromophenol blue, and glycerol. So the separation gel is prepared like this. Okay, so I told you, no, 1.4 molar tris HCl is taken, pH is 8.8. Okay, so these are the volumes of the respective components to be added for preparation of the separation gel. Whereas here you can see the concentration of tris HCl for the stacking gel is lesser, 0.5 molar and 6.8 pH, and the volume also taken is also lesser. So this is prepared separately, and they are used to they are prepared by pipetting it in between the plates. Okay, so what is the running buffer? Running buffer consists of tris HCl, glycine, and SDS. So the buffer tank should be filled with the buffer, and it is used for running the electrophoresis. It is called as the running buffer or the tank buffer. So what is the role of tris glycine and SDS? Okay, the glycine and the tris HCl, that is a chloride which is present in the tris HCl, they will help in the movement migration of the molecules in the electric field. So they are also charged, and they will help in the migration of charged particles in the electric field. That is the role of glycine and the chloride which is present in the buffer. SDS is also present, negatively charged SDS is also present. Now the staining will be done by using staining solution and destaining solution. First, after the separation of the protein, the entire gel is immersed in the staining solution containing all these three: glacial acetic acid, methanol, and this CBB at a particular ratio. So after the staining is done for 1 hour, and the destaining will be done. Then the destaining solution will contain glacial acetic acid and methanol, but the CBB will not be present. So destaining will be done for 24 hours. So the dye which is not bound to the protein will be removed. Only the dye bound to the protein will be visible. So this is how it is done. Okay, so suppose there are cells in a culture, you want to remove the cell, and separate the protein. The protein is taken and it is lysed. So by sonication and using detergents and by using SDS, okay, it is bound to the detergents and it is digested using the proteins sample loading buffer and is loaded onto the gel. Proteins are separated and it is visualized. This is how it is done. So these are the applications. Applications, separation of proteins, estimation of the molecular weight, and also peptide mapping can be done. Okay, so this is the role of this one. Okay, now I am stopping the sharing.